Hi, everybody. Hope you can hear me okay. I'm Tatyana, CEO and co-founder of ImYoo. Also graduated from Caltech with Ph.D. in medical engineering in 2021. And I just want to say I apologize if I need to pause, just recovering from a cold. David might have to jump in. So what we're building with ImYoo is a patient-enabled biodiscovery platform. What that means is we are trying to change in healthcare how patients interact with research. We want them to feel more empowered, more enabled, more engaged, and deliver more data to make precision medicine a true reality. And this is very personal to me because I had my own diagnostic journey. Started out in aerospace engineering field, and took a complete turn into medicine and biology because I really felt that was missing for me, and I really wanted to bring that enablement for everybody. And David is my co-founder here, so I'll pass it on for introduction to him.
Hi, yeah, David Brown, and I am the CTO and started this company with Tatyana Beck from Caltech. And I was in the Computation and Neural Systems program there, which focuses on machine learning and using it for novel applications. And uh, my background is computer science and ML, but I got pulled into the world of biology and bioengineering when I saw the data sets that were coming online, largely powered by sequencing. The amount of data that we can get today from small, non-invasive samples is kind of mind-blowing. And the idea of having that data coming from people all over the world to build longitudinal data sets and build more precise therapies is really exciting. And yeah, I don't think we can do it without machine learning. So yeah, I'm excited to tell you guys more about the data that we're building and what that can enable.
Okay, so just definition, longitudinal data means data across time. And right now, getting longitudinal data is hard and it's expensive. However, I think it's very important to get these critical time points across disease and across different types of people to really have precision medicine for everybody. I mean, if you really think about it, like when you go to a doctor or when you have clinical trials, it's usually just a few time points. You're just making decisions on those few time points. Whereas, you know, when you're feeling really sick, those are the times that get missed, or like we're kind of really missing this longitudinal picture of a person. Um, what we believe is that patients are the key to collecting that missing data. So here we have actually one of our patient advocate and patient advisor. We believe that if we give people the right tools to collect more data, we're going to be able to build medicines that are more precise for more people. And David, if you want to kind of chime in on the public benefit corporation angle.
Yeah, so we're incorporated as a public benefit corporation, which is a standard C Corp. And it just means in addition to uh, meeting the needs of our shareholders, is our stakeholders are also our patients. And in particular, our public benefit is making sure that patients are active participants in research. And we think this is really synergistic with creating valuable data because active, engaged people will stay in trials longer. They will give better data. They will be more open to sharing their hypotheses and sharing all the things that are going on with them. And we think that's the best way to understand biology is with them intimately integrated into the data collection loop.
And since we are changing this relationship between research and patient, we really had to innovate on how we're building the company. So we're mission driven and we have integrated expertise. So we engage the patients, we recruit the patients, we talk to them, and we also process the samples and we analyze the data ourselves and have AI kind of help us dig through this really rich data sets to actually pull out the insights. So we go from patient to the lab to all the way to analysis. And the mission that we're trying to support is building world's richest patient data platform, which can accelerate discovery for more personalized medicines. So we're building up ImYoo Atlas in partnership with our patient network. So we work with clinicians, we work with advocacy groups, we work with patient scientists. So patients who themselves have questions about their health. And we're enriching this data, and we are powering with AI models to really extract novel biomarkers and novel insights. And we delivered that to our biodiscovery partners, such as biopharma, diagnostic companies, and health monitoring companies. And we allow, and these people using this data can now build more biomarker targeted therapies for the patients. So this is kind of closing that loop in those between those three entities. So, and there are specific spots in the market that this addresses. So getting longitudinal diverse data is hard in clinical trials and studies, and that's one of the biggest things we're addressing. There's a large dropout of patients in clinical trials. There's a challenge, a struggle getting in good representation for diversity. And in terms of high fidelity, clinical trials that do have biomarkers are able to progress faster and are more effective. So in terms of longitudinal diverse component, we believe the at-home sampling solution that we're bringing, where patients can sample themselves at home on their own turf at their own time, makes it easier for them to stay in the study. And we've seen that with our own studies. And it's easier for more people to participate when there are less barriers. So that's what we're trying to address. And in terms of being able to get very high resolution data, we're using single cell RNA sequencing. We're going to expand to other assays. Being able to get really rich data from small volumes can help us get new biomarkers that we have not seen before that can accelerate these clinical trials and find new targets. So how are we doing this? So the technology actually comes out of Caltech. We've built a high fidelity multiomics data, and we have AI models that are powered by samples that have been collected for patients at home. So to do that, we have built a whole blood cell and gene expression preservation. So this lets us preserve the blood while it's being shipped to us, which expands our geographical access. Number two, we have developed a small volume. So we use this little, I don't know if you guys can see this, we use this little device here where patients self-collect blood and they send it to us, and we do high resolution assays on that. And we also build up an atlas, data atlas using our own platform. So what does that mean in terms of, okay, that's great, you have this technology, what products does it actually build? So right now, we're primarily working with therapeutic and diagnostic companies that are looking to find new biomarkers. In terms of products, we have the ability to execute discovery studies. So either from beginning to end, from patient to analysis, or we're somebody that you can add on to an existing study. And we also have built up our own atlas, data atlas, which people can subscribe to. So just to give you an example in terms of the discovery study, so this is the one that we're running right now. We were looking for biomarkers in inflammatory bowel disease and with patient-enabled sampling. So we started this in June, and we already have 35 patients across the country. So there's a bunch of different patients that are collecting their samples when they have a flare, when they're feeling quiescent. So really trying to understand this longitudinal component and give a molecular meaning to a flare versus it just being, kind of determined by, just reporting your symptoms. We're really trying to add this data component to this disease because that's really lacking. So I will pass over the torch to the describing the Immune Atlas to David.
Yeah, so on the subscribing to the Immune Atlas side, we have both a large healthy reference that we've built up. About half of our database is healthy reference, which is a nice cross-sectional view of what is the natural variability in the human immune system. And then we also have a smattering of cross-sectional across autoimmune conditions. And the longitudinal data that we're collecting so far is primarily in inflammatory bowel disease, psoriasis, and chronic fatigue syndrome. And people have been telling me that I don't brag enough, so I need to brag a little bit. The size of this database, about 400 people and 650 samples and 3 million cells. This is a massive data set. This is something that in single cell typically would take multiple centers across the country collaborating to pool samples from people over a very long period of time. And we've managed to do it with a team of 5 in about a year and a half. And it shows the power of being able to just rapidly spin up a study by distributing self-collection kits and getting those samples back to one central location. I think we're positioned to building an immune reference faster than was ever possible before, just because of this decentralized component. Can you go to the next slide? Yeah. And the reason that we're doing single cell, I got sucked into single cell during grad school, and I can't turn back now. Is because you get so much different possible outputs from a single assay. So here's just a few examples of what we can do from the small volumes that we collect. So on the left is we can use models that are trained on our reference database of cells to predict individual diseased cells in a sample, which could be used for possibly detecting minimal residual disease, or also just detecting early signs of disease where some subset of cells is misbehaving. In the middle here, we can zoom out and look at a person's entire immune system and build models on predicting different metrics. This is an example of an age predictor, where based on your entire immune profile, we can predict how old your immune system looks relative to the ages that we have. And then on the right side, the cool thing about single cell is you can also zoom way in. You can go look at very specific genes in specific subtypes of cells that are quite rare, and you can use them to differentiate potentially between different conditions. So we've got an example here of rheumatoid arthritis and psoriasis, which often have some treatment and some other overlaps, and sometimes can have trouble distinguishing based on existing markers. And we can see here that there's a marker that clearly separates just one gene in one of the 40 cell types that we have that clearly distinguishes between these two conditions.
So kind of zooming back out in terms of what's next for us? How are we thinking about growing the company? Where is it headed? And for us, it's everything we've developed thus far, this a decentralized way of collecting data, doing single cell RNA sequencing. It's all about helping us build our immune data atlas, specifically longitudinally. And right now, we're expanding our discovery studies. We're working with more partners to help them find biomarkers. As we commercialize our fixative solution, we're going to be able to access clinical trials and help out there. But for us, it's really all about, through all these partnerships, growing this data atlas, which will enable us to create our own assets, such as companion diagnostics, therapeutic targets. I think this data is really going to be critical for that. So kind of thinking about the accomplishments that we've done so far. So we raised a pre-seed about $2.1 million. And we've developed a solution that allows us to extend shipping time and lower shipping costs. We've secured partnerships with revenue over $200,000. We have shared IP across several partnerships. We launched our own decentralized study. Pretty sure it's one of the first ones out there where patients are collecting their own samples, and we're looking at what's happening across time in their autoimmune conditions. And we've also built a very strong reference database. Usually when you look at studies, there's just a few healthy controls, but we have over 100 healthy controls, really trying to understand that what is normal. So we got to a chance to really define that. In terms of what we're building up next, is we are going to generate a therapeutic perturbation data atlas. So specifically, we've been listening to our customers, and there is this growing need to understand, okay, why are some people responding to a drug versus not? Can we look at that across time? Can we say, like, what's a responder versus non-responder? What does that look like molecularly? Looking to expand our ImYoo kits into clinical trials. So this will obviously be a larger market for us and let us really say, okay, is our technology useful? And then we're also, in case anyone's in the audience, we're going to be hiring for business development as well as an immunologist to help us extract more insights from our data. And this will really help us expand our assays that we're offering and also help us scale the partnering. So, and just to kind of bring back, what does all of this enable? Like, every day we're motivated to build a future where we define diseases by molecular signatures versus kind of like the doctor looks at you and says, oh, you have this, because it looks like that. We now have the tools to really define it by what's happening at the biology, at the molecular level. So I really believe in the future, instead of saying, oh, psoriasis is you having itchy skin spots, it's going to be, oh, it's because your pathway is upregulated or downregulated this way, right? So we have the power to do that. Another one is patient treatment matching. I'm sure folks here have experienced this, going to a doctor and say, okay, try this drug, it won't work, try the next one. Instead being, hey, here's what I look like biologically. What should I be taking? How do I look like other people? What worked for them? And then three is understand, if we can longitudinally understand how a disease progresses, we can build preventative therapies instead of building therapies for when the disease is worse. So all this is why I'm really exciting about building this longitudinal solution for these patient-enabled sampling methods. And so thank you. And I'm really excited for you guys' questions. And I want to thank you for everybody who supports us: Caltech, Freeflow, Boost VC, Mars Bio, First Ventures. We were also part of Illumina Accelerator, as well as Johnson Johnson Innovation Labs, and LifeX Ventures. And I have my email there if anybody ever wants to talk one-on-one about what we're building. So thank you.
I wanted to kick off by, and the thing I always love just asking is a little bit about the history of when you guys first met, and then when you decided and how you decided that this was something that you wanted to take out into a company. Because I think that's, with scientific founders, that's, sometimes it's been part of the plan all along, and sometimes it's like, oh my goodness, I came across this thing and I want to maybe take that out and impact the world in a different way. So just curious, some of that origin story of the company would be great to hear about.
Yeah, for sure. Happy to talk about that. And usually, we talk about these high level aspirations, but reality is, starting a company is messy, and the pathways are usually kind of very curvy instead of straight lines. So David and I met actually in a machine learning class in undergrad in 2013. And this was at the time where I was going through my own diagnostic journey. I was actually in an aerospace program, wanted to be an astronaut, totally didn't expect to take this turn. But having had these healthcare experiences myself, I was taking this class and I met David, and we have decided, I think one of us asked the questions, if you had $1 billion, what would you do? And we actually landed on the same answer of building a company, or some sort of research effort to really help humans build biological solutions that will enable them to live longer, to do more, to be better. And ever since then, we've been really inseparable on that mission. We both worked at NASA JPL, and actually how we got into this is we volunteered at Caltech, at Richard Anderson's lab, as well as Viviana's lab, to learn biology. And we knew we wanted to start a company, so we quit our jobs and went to graduate school. And we worked on all sorts of different things. We didn't really know what we would end up actually making into a company. I worked on exporting toxic RNA from cells, AAVs, but it was really at the time of COVID, we were doing this project. We were trying to, do longitudinal data for Alzheimer's with a single cell technology. But I was remembering what we kind of did a proof of concept of this ability of doing small volume and single cell RNA sequencing. We're like, oh wait, the world just had COVID. Everybody's curious why some people get really sick, why some people don't get sick, right? So after we did a proof of concept, we saw that there's an opportunity in the market to kind of bring this data to other people, to enable better therapies, to enable better development of diagnostics. So that's kind of the journey that got us there. David, I don't know if you wanted to add anything to that.
Yeah, I want to add the frustration point, which I think triggered it in a lot of ways, was when we wanted to do this Alzheimer's study. We wanted to get blood, and we were like, blood's got to be easy, right? It's like you just poke a vein and you get blood. There's a bunch of people, like it's got to be easy to get longitudinal blood samples. And it wasn't. It was like you contact these blood banks, and like their version of sales for patients is they send you a spreadsheet that's got like 1,000 rows in it of individual, one person with Alzheimer's, 62 years old, and it's just like an aliquot of blood. It's like, it's not a person. It's treated like a an inventory of, of parts, and you have no way to follow up with the person. You have no way to ask them 10 years later, oh, did your Alzheimer's, did it progress? Did it stay stable? Like, what, did your drugs work? And then trying to do a longitudinal study with an academic center or a clinic was also quite painful. Like, it takes years to set up these collaborations and massive amounts of capital and grant money to just track people over a few years over time. And so I think this frustration point, that's when Tatyana was like, this can't be that hard. We got to be able to get our own blood, right? And she found this tap device. And I think that was kind of the pivot point where we realized that we were onto something valuable, because we were solving our own pain point as researchers at the time.
I have to add that Caltech is not really connected to a hospital, so that really enabled the solution. I think because Caltech is such a different environment where it doesn't look like other universities. I think if we were, say, at Stanford, this would have never came up because we were just gotten blood because there's a hospital there. So I think, it being in its own little island has really facilitated this approach.
Yeah, so as long as you're on the topic of the blood collection, maybe share, with the audience some of the performance metrics, volume, stability, results, and feel free to discuss mine in a very general way. Having now taken 3 blood samples, one at the world headquarters, the ImYoo world headquarters in the San Francisco Bay Area, administered by Tatyana and David, and then 2 by myself last week, 1 in each arm. And having been in healthcare and life sciences, really can appreciate the potential, implications of what I think you've done as a matter of necessity in order to advance the company, may in and of itself actually be quite valuable.
Yeah, for sure. Happy to talk about the process is, so a patient gets a kit, and they get this device here, and let's take the example of inflammatory bowel disease, right? It's usually you're flaring, you're dealing with your disease symptoms, and we ask folks to collect when they're feeling this way. So it's very empowering. So they take this little device here, they put it on their arm. There's microneedles here that puncture the skin, and it uses suction to draw the blood in. Typically, people can get between 0.1 mils to 0.5 mils, and we extract about 100 to 200,000 white blood cells from that. We can also extract specific cells. In one collaboration, we're specifically working on B cells. And what they do is once they've collected, they shake the tube, make sure we have something in there that prevents coagulation, and they send it back to us overnight. And in our labs, we process those samples, we extract the cells and preserve them. So that's kind of what it looks like. But David, do you want to touch a little bit on the metrics side?
Yeah. We see it's kind of like a linear drop off in terms of success at different volumes. So we see about 90% success rate of getting 200 microliters. 200 is more than what we need. We've gone down as low as 50 for our assay. But 200 gives us enough to do multiple aliquots and maybe take some extra for some other assays. But then if you want like 300, maybe it's 80%. If you want 400, maybe it's 70%. And I will be transparent in that 90% success is not great. If you had a 90% success rate for venipuncture, for example, that wouldn't work well in a clinic. Like, it's near 100%. And so we do give backup devices to everyone, so that if the first collection does fail, then the second one won't. But then also, I think, even with that, yes, if there's a few percent failure rate, like the logistical savings of not having to involve the entire infrastructure of phlebotomy and clinics is totally worth a few percentage points of sometimes people not being able to do a draw.
Can you shifting topics a little bit, can you maybe dig into or explain, because I know that your background is also heavily, computer science as well. So can you explain how kind of computer science tooling, including, the buzzwords of the day, like machine learning and AI, of course, but just other tooling, like how are you melding that with what you're doing, and what does that result in, essentially?
Yeah, there's two big areas that I could touch on. One is kind of I think it's better now, but certainly 4 years ago, I would say biotech was not getting enough computer science love. It was like software and infrastructures were not being built to accommodate data at the scale that it was coming in a convenient way for researchers. So a lot of the stuff that I do for the infrastructure of ImYoo is making us able to analyze data very quickly. So just as like a tangible example, these gene count matrices that we get back, we get gigabytes of data from one of these small samples. And previously, if you wanted to collate data from hundreds of people together into a single analysis set, that requires loading hundreds to thousands of files spread across servers, and can take a bioinformatician it can wipe out their day of just like finding everything and putting it together into a single thing so that you can work with it. And that was, we couldn't work in that environment with the rate that samples are coming in. And so applying computer science principles to these problems, there's a lot of optimizations that can be made in terms of database design and data access, so that we're at the point now where if we need, let's say we want data from everyone who has RA, we hit our API with a query that says, give me all the cells from people that have RA. It goes in, it finds, it's been indexed, so it finds all that data from across all the thousands of files. It pulls it together and it delivers it to the bioinformatician in a matter of minutes, as opposed to what might have been a long time. So I think there's a lot of like little optimizations like that can help with kind of big data biotech. And then on the machine learning side, it's kind of funny actually. We've been doing some kind of coaching on our pitching lately, and someone asked us, I mentioned casually something about AI that we were doing, and previously AI was not in our pitch at all. And when they asked about it, they were like, well, wait, you're doing AI stuff? We were like, oh yeah, we do AI stuff all the time. That's like, how else would we do our analysis? That's like the bread and butter of our analysis. We train models on cells, we train models on people. And they were like, well, you should talk about that, because there's a lot of people that maybe you could theoretically talk about AI, but maybe you're not incorporating it in your day-to-day. But our data sets are such that we can't do it without machine learning. The cellular states are all defined based on machine learning models that are trained on our reference database. And it's just, there's too many cell types, and they're being discovered faster than people can manually annotate and identify them. So it's a critical component in the workflow. And yeah, I think it's a really exciting time for what these models will be able to do.
So next question is the idea here that any doctor anywhere, not necessarily a specialist, can take a blood draw of their patient and access your database to diagnose the patient's disease? Will you charge a fee for this service? Also, you may be able to suggest what works for other patients' symptoms.
Yeah, I think that's something that we're working towards in the future. Definitely not somewhere where we are now. We are working, we have some pretty excited clinicians. We're excited to send back their patients with our kits and learn about their disease and research it. But right now it's very much in the research phase. But I do envision in the future, as I said, like when our Immune Atlas grows big enough, once we certify some of our essays and some of our findings, we do think we're going to be able to do that, which will be really, really cool.
One other question, David, just to sort of drill down on the ability to take a sample when there's an acute flare-up, right? Because immune disorders are characterized by flare-ups, attacks, symptoms just sort of cropping up rather quickly. And so presumably getting that sample at that point in time with your device, where you can do it literally at the moment, provides a huge benefit that is absent in basic clinical trial design today. So my suspicion. Maybe you could comment on that a little bit more.
Yeah, I haven't seen any clinical trial designs where they incorporate a patient. Usually, if you're not feeling well to a point where you need to be taken to a hospital, you just go to a hospital. But no, there's no system in place that I know of where a patient can be like, there's something wrong with me. I want to take a data and that data be, heard. I haven't seen anything like that.
Yeah, there's a quote that sticks in my head that we heard from a director of translational research at a big pharma company when they've been talking to us for a while, and we asked them about who else has data like this, like who else, what's the market, what's the landscape, who are we competing with, and their response was, data like you're collecting just doesn't exist. And I was like, okay, I mean, pros and cons to that. It means that we need to prove out a hypothesis that this new modality of data is valuable, which is
right.
what we hope the IBD study shows. So that's a con, but on the pro is if it does pan out, then we're uniquely positioned to seeing things that nobody has seen before.